Like to welcome everyone. I'm Dr. Suzanne Buchanan, Executive Director of Autism New Jersey. Thank you so much for logging on today to learn more about the causes of autism. We're looking forward to having a great conversation moderated by our own Communications Director, Stacy Sherman. And in the interest of time, I'm going to hand it right over to you, Stacy. So thank you so much.
Thank you. Hello, everyone, and welcome. Last year, Autism New Jersey hosted a three-part webinar series on the causes of autism, bringing together experts in brain development, genetics, and environmental science. The response was remarkable. More than 850 people attended, and more than 600 follow-up questions were submitted by families, clinicians, educators, and advocates. What became clear from those questions was that there's still a strong need for clarity. Families want to know how different scientific perspectives fit together, how to interpret risk responsibly, and how research findings apply or don't apply to individual children. Today's webinar is a direct continuation of that conversation. We've invited back the same three Rutgers scientists who anchored last year's series. Dr. Emanuel DiCicco-Bloom, who studies early brain development, Dr. Chiara Manzini, whose work focuses on the genetics of autism and neurodevelopment, and Dr. Alycia Halladay, Chief Science Officer at the Autism Science Foundation, who specializes in environmental and population-level research. Our goal today is to clarify what the science supports and to help families think about risk, development, and differences with greater confidence and less fear. Let's start with how autism emerges. Dr. DiCicco-Bloom, when scientists say autism involves differences in brain development, what does that actually mean?
Well, in fact, the brain starts to emerge in utero during gestation. The nervous system begins to fold over at 3 to 4 weeks of gestation. So we're talking about the brain development. We're talking a great deal prenatally as we build the cells of the brain. Those are neurons and glial cells and other cells, and how those cells begin to lay pathways to connect one nerve cell to another to another. So a lot of what we're talking about occurs in utero, and mother's experience and mother and child genetics play important roles in building the brain.
Okay, parents often hear that autism is highly heritable. Dr. Manzini, what does that mean in practical terms for families?
So thank you, Stacy. So heritable means that something runs in families to like make it understandable. There is something in your DNA that is inherited through the generations. And when scientists started trying to figure out whether autism was genetics, the first thing that you did, in the 70s and in the 60s was to look at identical twins, because identical twins is one embryo that splits in two. It's like two almost identical DNAs. And in that case, they found that in up to 90% of cases, both twins had autism. And that told us that it was genetic. There have been many, many genetic studies in the past, like 40 and 50 years, that have expanded on this. Now we know, for example, that if you have a child with autism, you're more likely to have a second child with autism than the general population. But also we identified hundreds of different genes and genetic causes. And one of the things that I think is very complicated for the families is that sometimes you have one single genetic cause that by itself is able to cause autism, but you could also have a situation where you have a predisposition. You can think about this as diabetes. You know that a grandparent or somebody in the previous generation has diabetes, that you have risk to develop diabetes, but you don't know if you will, have that particular disease. And so both can be true for autism, and maybe we can expand on that a little bit later.
Okay, thank you. Dr. Halladay, let's bring you in. When researchers talk about environment in autism studies, what does that term actually include?
So I think when researchers talk about the environment, they're very, very broad-based in what they consider, right? So they consider things like not just the air we breathe and the water we drink, but they also think about access to services, the neighborhood that you grow up in, medical conditions, exposures prior to pregnancy, exposures during pregnancy, perinatal exposures, things that happen during labor and delivery. I think when the community hears the word environment, they're used to hearing the word environment when it comes to things like climate change or wildfires or medical procedures. So I think that scientists can be very broad about it, but I think the community kind of probably needs to also be a little bit broader. But with that comes new ways of measuring the environment. Some of these environmental exposures, and we'll talk about this, are things that we don't even know we're exposed to. And then some of them are within our control.
Okay. If multiple influences are involved, timing becomes critical. So Dr. DiCicco-Bloom, at what points in early development do autism-related brain differences seem to emerge?
Thank you for that question, Stacy. So this actually builds on what I said earlier about early development, a lot of it occurring in utero. But different systems are developing at different times. And so the differences in numbers of nerve cells may be over by the middle of gestation. But those cells have to move and make connections. But once we are born and we have those differences and connections, we experience the world and we grow synapses where two cells connect to each other. The synapses are just being established at the end of gestation and during the first early years during childhood. And so a lot of building of the brain occurs not only by starting with the cells and circuits, but the influence of incoming experiences, whether it's with parents or environmental experiences. If you're living in the middle of an urban city, there are different kinds of experiences than if you're living on a farm. And so these different substrates then are influenced during early childhood. I think it's reasonable to imagine that by adolescence, the building of the brain is probably done, although we'll see. But a lot of what goes on then is modification. Certainly, as many parents know, puberty is one heck of a modification of the ongoing child you had. And so different stages for different processes.
Okay. You lead very nicely into my question for Dr. Halladay. So why does the timing of an exposure matter as much as the exposure itself? Oh, Dr. Halladay, we can't hear you.
So sorry. If you can think about what Dr. DiCicco-Bloom just said and how there are different stages of brain development and cells are born at one stage and then they connect with other neurons at other stages and then glial cells or support mechanisms develop at other stages. Environmental factors target different times or different parts of that brain development. So I'll give you an example. For air pollution, we see effects, very small, but still there, effects on the probability of having a child with autism if the mother is exposed during the second or third trimester of pregnancy. On the other hand, the effects of folic acid, which is a prenatal vitamin, given all women should be taking folic acid during pregnancy, those effects are best seen if you've taken that either prior to conception or around conception. So it just depends on the way that a particular environmental factor works in the brain. And that could even tell you, what you should be looking for. And that's why science and research is so important, because it can even dissect out, okay, well, this is what's happening in the brain. This is a particular exposure of interest in what it does. And do they connect?
Okay. And since genes set the stage early on, Dr. Manzini, you mentioned earlier that autism can be caused by changes in one gene versus many genes. So why does that distinction matter?
So it matters because a single gene versus a multi-gene, that it's called the polygenic risk, are actionable in different ways, and also are sometimes understood by the families or by the autistic individual in like different ways, right? So if you have a single gene or a single genetic cause, you know that that will give you 100% risk of leading to autism. So you'll know that that's the one cause. And sometimes the one cause is important to know. It also can generate community because you can meet with other people that have the same condition. And also, if once that you understand how the gene works, you can start thinking about therapies. And now there is a lot of talk around gene therapy for some specific genetic causes of autism. But those only account for around 20%. And often the more severe end of the spectrum. Then there is this polygenic risk, which means there are multiple genetic changes that you inherit from your family that add some genetic risk. And each one is a very small percentage, and then all of them together. And some of the audience might have seen this description of polygenic risk scores or PRSs. They're being developed for all kinds of polygenic conditions, diabetes, heart disease, polycystic ovarian syndrome. And it's kind of like the odds that you will possibly develop a condition. And so it's very difficult for the clinical community now to figure out how to report them to people, because it's not going to be, oh, this is it. Like, it's just telling you this is possible. But these polygenic risks actually can interact with the environment. And this is what Alycia was saying before. You may have a predisposition, and then specific environmental factors come into play. And there is a research really now starting to put these together, which is very important.
Thank you. So families ask often why there isn't one simple explanation. So, Dr. Halladay, why is it so difficult to prove cause and effect when studying environmental factors in autism?
Well, yeah, I can completely understand all the frustration, but think about all the different things that go into that cause and effect. So one, you have to know, autism is a very heterogeneous, we term it now autism spectrum disorder rather than just autism, because it encompasses a wide spectrum of individuals. So someone, an environmental factor may lead to one certain feature of autism or a group of features of autism that doesn't look like somebody else with autism. So that becomes confusing. There's also the idea that nobody really knows. You can't really study gene environment interactions most of the time when most people don't know their genetic makeup, right? And that's not their fault. Like, not everyone is given the option for a genetic test when they're born. And then the third reason is that we don't even know as individuals what we're exposed to most of the time. So we could probably tell you what medications we're taking and the dosages. Hopefully most of us can do that during pregnancy. That you can probably count your steps. You can probably think of a lot of different environmental factors, but I don't think you would know how much chlorpyrifos you were exposed to, or what air pollution level you're exposed to on day 130 of your pregnancy, right? So most people don't know themselves their environmental exposures. So it's very hard to somewhat monitor them. Scientists are getting better at that. There's a whole new field of research called the exposome, which is to look at all of the different things in the environment and how they're affecting the body, and then putting that information together. But it is very hard to do a cause and effect because we're influenced by so many things at once, most of which we don't even know about. And then also autism is such a broad-based diagnosis. It's a spectrum that, what may reflect a gene environment interaction in one person may not in another person.
And genetics presents a similar challenge. Many people expect a single autism gene. So, Dr. Manzini, why are so many different genes linked to autism?
Exactly for the reason that Alycia was just talking about. I mean, the current definition of autism spectrum disorder became a diagnosis in 2013. Before, it was split in several different disorders, with Asperger's
Uh-oh.
as the least, the people that were the least affected, and autism as one of the most severe. Now it's all in a spectrum, and it was helpful at the time because none of the diagnoses that were used at the time were fully able to help us understand autism. But the genetics that has been done on the autism spectrum disorder community has really shown that the genetic causes can be different. And so that you have these more severe genes for these developmental disorders that often have intellectual disability, developmental delay, a variety of things that now have actually been defined as profound autism. And then the polygenic causes tend to be more in the other end of the spectrum, but there could be everywhere. And so, really trying to figure this out has taken a very long time. And there are many genes because the development of the brain is complex, the function of the brain is complex, and a lot of genes that tweak that function a little can lead to one of these presentations that we call autism, which in reality is a spectrum of different conditions.
And the brain adds another layer. Dr. DiCicco-Bloom, why can't brain scans be used to diagnose autism?
Well, when you think about different parts of the brain and their functions, excuse me, you need to think about how it could be altered. So let's take two large extremes. A brain that looks typically formed. We've got the frontal lobes that allow me to plan and get here on time, parietal lobes, which integrate sensory experiences, the occipital lobes, which allow me to see the world, and then all the connections between them. So grossly, we've all probably seen MRIs or CAT scans, and you can kind of see obvious differences like, oh, the right-hand frontal lobe is smaller or had a stroke. Or the lower part of the brain, the cerebellum, is much smaller than you'd expect for that person. So those are easy things. But one of and as clinicians and practitioners, we kind of know the different regions and what they're structurally what these structures do. But many brains, the majority probably, look typically built. So then the question is how do they function? And so, as you're aware, radiologists have done structural analysis, and there is no obvious difference, but a lot of neuropsychiatric disorders, the brain doesn't really look structurally that different. There might be variations, but nothing that's definitive on a brain scan. So then there's the next level of analysis is functional connectivity. And so how does one region communicate with another? For example, when I'm speaking right now, you may be seeing me and you're also hearing me. These are two types of sensory inputs, and we can ask how that information gets from the beginning, the ears and the eyes and the lower part of the brainstem, but then it's got to project up to the temporal lobes, the occipital lobes, and so functional differences. But there's no clear signature of a functional difference that says that's autism and not early Parkinson's disease or Alzheimer's disease. And so it isn't definitive. Pathology, looking at a brain under a microscope, can diagnose some disorders like Parkinson's disease, but in fact, pathology cannot tell you that a person has autism. Nor can it tell you by looking at it grossly whether there's epilepsy. And so these are challenges because we don't really have the level of analysis related to those functions in brain scans, whether they're structure or function.
Okay, that leads into our next area. We'll look inside the brain. You already talked about brain connectivity. Connectivity can also be shaped by early experience. So, Dr. DiCicco-Bloom, how does early experience interact with brain development in autistic children?
So let me build on this idea of both connectivity and early experience. So I probably already started this conversation a moment ago, but if we think about a circuit, let's take the eyes. Information goes into the eye, and there are receptors that see light. And then those nerve cells, the receptor nerve cells, project to a region called the thalamus, at which point it makes the connection of the eyeball nerve on the retina to a thalamic nerve cell. And then that nerve cell projects back to my visual cortex. Now, if you've got billions of nerve cells doing that, it is a lot of work, and it's also a lot of analysis without putting an electrode in a nerve to measure its activity. Everything is derivative. It's not cell type specific. So as we're building, so let me get to now, that's the idea of building circuits with multiple components, early, middle, and late. Now, let's talk about having a brain that's different, maybe in the numbers of nerves or the connections between nerve cells, as we just talked about. Well, our brain is not static. We are constantly experiencing the world, and we're learning. Think about a young child. You're showing them the alphabet and saying, "Hey, apple." Even us old folks are learning from experience around ourselves and modifying the circuits that are there. So if you have different nerves and different connections, when those experiences come, you might experience them differently. Let me talk about a real life situation. If you have a child on the spectrum and they're in a loud and complex environment, like going to Costco, for example, noise and incoming information, well, for one individual, they may be okay if they're holding a parent's hand or holding their toy or wearing their earphones. But for another, you might pass their threshold. And at that point, the system says escape. And that escape could be withdrawal, hiding in the corner, wrapping up in a ball, holding onto a leg, or the opposite, instead of going inward, going outward with a behavioral outburst, which is challenging for all, including the child and the parents, as well as those in the environment. So having differences means that common experiences will impact that child differently. And consider what the Eagles Autism Foundation has done, for example. There are sensory rooms now at football stadiums because typical excitement at the football game can be overwhelming.
Thank you so much. Dr. Halladay, question for you. How should we think about maternal health? Is it environmental or biological?
Yeah, I think that that's a really great question and something that we need to better understand through science and research. There are likely genes that are affected in autism or that vary in people with autism that also vary in other conditions. As a matter of fact, we know this to be true with certain things, but for something like, for say, gestational diabetes, so there seems to be this persistent link between a mother having gestational diabetes and a diagnosis of autism in their child. Is that because there's a shared genetic factor, or between autism and gestational diabetes, or is it because the high sugar levels during gestational diabetes that seem to be a little bit less controlled than if you had like a type 1 or a pre-existing diagnosis? Is that the factor that triggers autism, or at least contributes to autism in the child? So I think that there still needs to be a definite kind of thinking about that. The same could be same for prematurity. So what are the underlying causes of a baby being premature? We do know, and we'll get to this, that premature infants are 4 to 5 times more likely to have an autism diagnosis. Is there a shared underlying biological effect? Or is there something, specifically about being born too early? So I think that depends on the condition, but and we need more research around that, but I can certainly understand why that's a confusing point.
Adding to that, even within the same family, development can look different. So, Dr. Manzini, why does genetics help explain? What does genetics help explain about why siblings can be so different?
Well, Stacy, if you think about, siblings, right? If anybody in the audience has siblings, there are things that look like your mom, things that look like your dad. I have a whole things that look like my grandma. And so the different things that define how you look, your health, your height, every single aspect of your body can be inherited through the genetics in your family. And so what we know so far is that there is a baseline genetic risk for psychiatric disorders. And then, for example, in these risk factors, the polygenic risk factors, autism and ADHD are overlapping. And you could have one sibling that has more of the ADHD component and another that has more of the autism component. And then this can compound with the single gene mutations, because there have been studies that have shown that even when you have the one sure thing mutation that you know is going to cause a form of autism, there is still the background genetic risk that can add up. And so you could have one sibling that has more of that and is more severe, and a sibling that has inherited a little bit less of that additional genetic risk and be less affected. And so it's really the general distribution of what we call genetic traits that are what defines how we develop. And then the environment can also compound on that. Every pregnancy is different. Different things can happen with different children. Early development can be different with different children. And so that, I think, helps explain the variability.
Okay. Zooming out to the population level, Dr. Halladay, how do population level findings differ from what they mean for an individual child?
Yeah, that's a great question. And it goes right back to what Dr. Manzini said, which is that there's a lot of different genetic variation. You take a group of people with autism, right? So autism spectrum disorder considered broadly, and you have different levels or different features of autism within that broad diagnosis. And then on top of that, you take a group of people who are genetically kind of distinct, right? So some of them may have different background genetics. Some of them may have predispositions to other things. Some of them may have single gene mutations they don't even know about. And then even the polygenic risk scores. And then you try to isolate one environmental factor. It's kind of a miracle that we're finding anything at all. So it does speak to the strength of some of these environmental factors. But when you look at a group of people with a wide range of deficits and strengths and challenges, and then also factor in all the different ways people are different, you can take that population level risk and you can kind of think about it in terms of, okay, well, this is a baseline for me to think about, but it should never be used as being predictive, right? So you can say, I'm going to have a premature infant. The odds of having autism are, say, 1 in 10 or something like that, rather than 1 in 31. That doesn't mean you're the one. It doesn't mean you're the 9 that don't have it, right? So you just kind of have to use those numbers, those population numbers, and keep an eye out and keep focused on supporting early intervention and early support for that child.
Studies often report small changes in risk, which can be hard to interpret. So, Dr. Halladay, how should families interpret these small changes?
So, the small changes can be very important. And sometimes, as with the population level numbers, even with the small changes in risk, these are very, very helpful for scientists to take that information and say, oh, well, this may be a particular mechanism, or this may be a pathway of interest, or look at the way this particular drug works at this particular period in time. What's going on in the brain, and does this particular pathway make sense? Should we study it further to look at ways to ameliorate the detrimental effects of autism? So just as much as they're important for each person, they are also important for scientists. But when you see a small population level change in probability of a diagnosis, I would definitely pay attention to that, because all of these things can add up, as Dr. Manzini said. You know, all these different little things that are associated with autism. You may have there may be a small risk here, and a small risk here, and a small risk here. I think this all leads to trying to mitigate environmental factors that are known to be associated with autism as much as you can. And these include things like air pollution and pesticides and prenatal birth. Everybody wants to reduce prenatal birth. We shouldn't stop doing that. But also be aware and be prepared for a wide variety of outcomes should there be that exposure.
Okay, thank you. To connect early brain development with real-world differences, Dr. DiCicco-Bloom, how do early brain development differences connect to later communication or social differences?
Well, that can be a challenging question. So we have spoken already about building a brain, which might have different kinds of nerve cells or different locations. Those nerve cells may not be in their right locations to make connections. And then if we have a different brain, as we already discussed, different experiences, whether they're environment, whether they're genetic predispositions, or whether they're directly experiential emotional experiences, they will alter the maturation of that brain differently in each of us. Keep in mind, there are those of us who are artists, those of us who write, those of us who are basketball stars or ice skaters. We bring with us an inherent capacity based on environment and genes. And so to try to draw a line from the way we're built to later communication and social interaction is probably a bit of speculation. On the other hand, I think we've all heard about one of the emotion centers of the brain called the limbic system, and the amygdala is one that's frequently seen in the newspapers. It's either too active or not active enough. 25 years ago, when functional brain scans were done on individuals with autism, mainly adolescents and adults, they noticed that the amygdala, a center which basically tells us this is a good place to be, or this is a fearful place, a dangerous place, they found that individuals with autism seem to activate that place less. And so there was a concern, and in fact, pathology, counting nerve cells in the brains of adults who counted the number of neurons in the amygdala, and they were less. And they said, oh, this is failed development of the emotional sensing center. And that's why there's less connection to social experiences. But then, as we got more sophisticated about doing MRI studies with little picture shows to show faces that were neutral, or faces that were scary, or faces that were happy, and we looked at the amygdala, whether or not it lit up more when it was scary or more when it was happy, they found out that individuals on the spectrum weren't looking at the faces and the pictures. One interpretation, and certainly individuals reported, that looking at the scary face made them feel so bad they couldn't look. Now, we're talking about a high-functioning individual who's actually able to communicate and perceive and report their emotions. That's a very sophisticated individual, and in those days, they were probably individuals with Asperger who would report about it was scary. In fact, Temple Grandin, when she's a well-known autistic adult who is a environmental engineer, she would have no problem talking to a large audience, but she basically shunned a one-on-one conversation. It felt badly. I think we all can kind of relate when we feel threatened or anxious or out of control when faced with certain individuals or experiences. But you see, now we're thinking about behavioral things, and we can't really take a behavior and pin it back to a specific brain region. The amygdala was an example of where there were differences, whether it was the numbers of cells there, whether it was activated in a typical way, or there was other avoidance behavior not to be activated. And so trying to make a one-to-one relationship between brain development and adult communication and social experiences is difficult. And that's why, kind of harking back to an earlier question, we can't look at the functional signature of an individual's brain and make a diagnosis of autism and report where that's occurring.
Okay, thank you. These differences, these brain development differences, unfold within both biology and environment. So, Dr. Halladay, which environmental factors have the strongest evidence of being associated with autism risk?
Yeah, so there have been a few that have been studied over and over again and have consistently shown an effect. One of them is the taking one particular anti-seizure drug called valproic acid during pregnancy. I don't want people to stop taking their anti-seizure drugs. Valproic acid seems to be one that's particularly bad, and it actually has a black box label now, so no pregnant women should take it. However, there are other anti-seizure medications that a pregnant woman can take, so I don't want people to just stop taking their anti-seizure medications. Another one that I mentioned is premature birth. Now, again, we still need to do more research on whether it's an underlying genetic factor or some underlying biological factor, or whether or not it has something to do with the fact that a baby is preterm and the brain hasn't fully developed yet, and they're being exposed to all sorts of things that they wouldn't have if they were still in utero. Another one that pops up over and over again that's been consistently associated with autism is maternal illness. So if a mother has an illness during pregnancy, particularly one where she has a fever, that child has a higher probability of an autism diagnosis. Now, this is important to know because there's definitely things that women can do to avoid getting sick. Get vaccinated during pregnancy, wash your hands, stay away from other sick people. So this is one that I think an environmental factor that I think a lot of healthcare providers can get behind the same kind of recommendations for everyone. Another one is air pollution. This one is a little bit harder because we cannot control our levels of air pollution. Sometimes we are exposed to high levels without even knowing it unless we cough or can notice it. Another one is parental age. Over and over again, both the increasing age of the father and the increasing age of the mother seem to be, those children have a higher probability of an autism diagnosis. Now, that being said, there is some evidence now to show that that is due to genetics, to some factors. As women and men get older, both their sperm and their eggs, the DNA changes ever so much, enough to cause a neurodevelopmental disorder. So that might not be environmental, but for now I'm putting it on the environmental list. And then there are things that very preliminarily, we still need more studies on this, might actually promote resilience from an autism diagnosis. So taking folic acid, especially during conception or early on during pregnancy, seems to lower the rate of autism in women that have taken it at the right time, that being that very early phase, which means you have to take it every day even before you know you're pregnant. Those studies need to certainly be replicated and extended because they've only been done in a small group. But there are things, and there are things. These factors that I mentioned have been shown over and over and over again in multiple studies to produce various levels of probability, but still probability, a higher probability.
Got it.
I was wondering if I could ask Alycia a question. This is unscripted.
Yes.
So,
That's okay.
um,
Mhm.
50 years ago, when I was a kid and I watched my parents filling their car with gasoline, there were reports out of Chicago, very powerful reports, that environmental levels of lead, which could be measured in the city, on the ground, in the blood of children, correlated. The more lead, the lower the IQ of the children population,
Hmm.
individual, but as a population. And this, remarkably, this environmental factor, which seemed to be bad, the government said, we're gonna get rid of lead. It was part of the catalyst to make gasoline more effective. And they got rid of lead. And then subsequent studies showed reduction in the environmental lead, reduction in the blood levels of lead of children, and improvement in the IQ of the entire city. And I think there have been studies not only of this metal, but other metals in the environment. They kind of raise the threshold for many disorders, and any developmental disorder, intellectual disability, and autism. How do we view those kinds of ubiquitous natural components of our environment or industrial waste?
Yeah, so I also want to emphasize here that some environmental factors that are associated with autism are also associated with a number of other different factors. Take example, some of the pesticides that certain environmental groups are trying to get outlawed, like chlorpyrifos, for example. Those have been linked to autism, but they are also linked to things like lower IQs. They're linked to cognitive disabilities. They're linked to fertility problems. They're linked to cancer. So it's not as if there is an environmental factor that we know about that is just linked to autism. Valproic acid, as I mentioned, that's linked to birth defects, right? So cleft palate and other, even more severe birth defects like spina bifida. So it's not specific to autism. And I think that we should probably use some of these lessons, the lead lesson, which is a triumph in public health. If you can think about the amazing things that have happened in public health over the past 50 years, the elimination of lead has certainly led that charge. But we do need to think about not just the effects on autism, but the effects on broader neurodevelopmental issues, right? So IQ, ADHD, autism, other developmental disorders, because elimination of these toxic chemicals, which we know are toxic, can improve the overall health of the entire population and not just, say, change the prevalence of autism. So I think lead is a good example. Another example is mercury. So mercury has not been associated with autism. It was hypothesized to be associated with autism. It's not. But that doesn't mean that we shouldn't get rid of methylmercury levels in the environment, because methylmercury, which is different than ethylmercury, but methylmercury, which is what is found in fish, and it comes from coal burning power plants, dumping things into the water, into the ocean, then the little fish eat it, and then the little fish are eaten by the big fish, and things like tuna and shark accumulate methylmercury, and you eat it. It can be incredibly toxic. And autism is actually probably not even the worst thing that can happen to you if you eat high levels of mercury. So I think that this is a lesson that even if it doesn't change autism, or if it's not associated with autism, there are also environmental factors or environmental things we should be getting rid of. We should be getting rid of chlorpyrifos. We should be getting rid of smelt from coal burning power plants. These are all things that we should be doing. And that would be a public health miracle if we could change the developmental trajectory of kids in general outside of autism. So I'm standing off my soapbox now, but thank you for that question.
No, very important question and answer. Thank you so much. Let's turn to understanding autism through multiple models. Animal studies help test these ideas. So, Dr. DiCicco-Bloom, what can animal studies teach us about autism, and where do they fall short?
Well, obviously, we can't do brain developmental studies on children, on humans. And so we can certainly look at the effects of medications once we've learned what medications might be effective. And so animal models, whether they're non-human primates or rodents or worms and flies, we can we can do initial tests of hypotheses. So we can ask questions of regions that I alluded to. For example, how about forming the front of the brain or the amygdala or the visual cortex? So animals allow us to ask very targeted questions in controlled conditions. And as scientists, we come up with reasonable ideas, and at other times we come up with totally crazy ideas. And that actually is a good thing because we can't be conservative in thinking we know everything and there everything follows. Sometimes we have to come up with a counterintuitive hypothesis or look at the results we get, which don't fit our idea of what's supposed to happen. And so animals allow us to specifically target a region and then reproducibly demonstrate to ourselves that it seems to be part of the process. In fact, it might be necessary for the process. And so if it's not there, you don't get the bad outcome. Also, the question is, if it is there, it is definitely a cause. We call this either being necessary or sufficient. And so I'm sure my colleagues will talk further about this, but we do this routinely with genetics. We ask about a gene in an animal that is helpful. Flies and worms can be very good about finding genetic circuits, and they're very quick and inexpensive and reproducible. But then we need to ask about something as complicated as a vertebrate nervous system, like a mouse or a rat or a primate, and ask about whether or not my belief that the amygdala is really important for social function. And I will tell you, this was a very powerful and important area of research 25 years ago, where not only rats and mice, but even primates, monkeys, had their amygdalas removed. And lo and behold, the animals, particularly the primates, didn't have the obvious autism that was predicted. And so that's really important because it then tells us our idea was reasonable. The test was done rigorously by several groups. And now the information is it's not just a one region, even though I can point to different regions that when I hurt them, something gets broken. It doesn't mean that region alone is the cause of it. A lot of current thinking about cognition, not an area that I study, is that it's really the network. It's everything acting together and modulating moment to moment. So there's no doubt if I have a stroke in my left hemisphere in my language area, I'll lose language, and then I will try to adapt. If that occurs, by the way, in a 1.5 to 2-year-old child, they will lose language, but 2 years later, they'll be speaking again because the brain can be plastic. It can find other pathways. In an early childhood, a child who loses language due to a stroke can regain it on the other side. And so those are natural experiments, but we can't do those. And so animal models are very, very helpful in allowing us to ask about the importance of a factor, what regulates it, and then gives us a foundation on which we can then try therapeutic interventions by increasing or decreasing the activity of different nerve cells, for example.
Okay, thank you. Rare genetic conditions also offer insight. So, Dr. Manzini, I saw you nodding a lot, and give you a chance to weigh in here. But how do rare genetic conditions linked to autism help scientists understand more common forms?
Yeah, so thank you to Dr. DiCicco-Bloom for setting everything up, because basically that's mostly what we do in the field of studying rare genetic conditions, is make animal models, right? And you first need to figure out what the specific change does. In some cases, it breaks the gene. In some cases, it changes the function of the gene. So that's the first thing that you need to figure out. And you usually do that in an animal model. And then you have to figure out what the gene does. In a lot of cases, you find the gene through a genetic study, and you are not you're not really sure of its function and how it is related to development of disease. And so you also have to figure that out. And there is a playbook to do that in science right now. And is relatively easier. And I know it doesn't feel fast to families, but it is definitely faster than taking all of these multi-gene causes that we're not 100% sure what they do and look at their interaction and trying to figure out, if you already don't know what half of those genes do, trying to figure out how they act together and how they're modified by the environment, for example, can be very complicated. So you start with the rare genetic conditions. And then you can figure out, are there specific changes in the brain or in the blood, right? That's what we call biomarkers. Sometimes it's something that helps you determine the status of a disease, or whether somebody has something, the easiest thing would they check your HDL in your blood, like, you know. Um, and so can we see if the same changes that were found in a rare genetic cause hold true for the more common forms? And this can be very helpful, because we were saying there's this large kitchen sink of the autism spectrum disorder, and there is a lot of research now trying to find subtypes and trying to figure out if subtypes are actionable. So you may find through a rare genetic condition a particular biomarker that is also present in a subset of common forms of autism, and they all respond to the same treatment, right? So we're kind of dreaming of a day like that, where everybody can have their personalized treatment. Because again, there's not going to be a smoking gun. There's not going to be one treatment that's good for everybody. We really need to figure out how to treat, or how to address the specific subtypes.
Okay, thank you so much. These insights, they also clarify common misconceptions. So, Dr. Halladay, is there an environmental hypothesis about autism that has largely been resolved but still persists publicly?
Yes, there is. And I think it's no secret to everybody who's watching and listening what that is. It is vaccines. So in the late 1990s, there was a paper published that suggested, didn't demonstrate, but suggested that there may be a link between the MMR vaccine and an autism diagnosis using a very, very small sample size. That paper has since been retracted because of concerns about how biases by the investigators, and then also concerns about the study design. But after the original study was published, the autism community and even the bigger, young parent or new parent community was incredibly worried about the thought of their child getting a vaccine that was supposed to prevent a disease, then causing a disorder like autism. And there was somewhat of a panic in the community. So understandably, both the United States and countries outside the United States focused efforts onto using new data, existing data, population level data, cohort level data, all this different types of data, kids that are at risk for autism because they had a sibling, populations, larger populations, groups of people monitoring very carefully their exposures, which resulted in literally hundreds of millions of dollars over the course of decades of research being done. This includes data in the United States, in Scandinavia, in Japan, in Mexico, all over the world, Europe, Israel, to show that there has been absolutely no link between vaccines and autism. However, this theory persists because when you go to a pediatrician's office, it's not the most pleasant experience in the world. And you go in and your child is getting a shot, and that means they're going to cry and you're going to have a cranky infant. And the whole thing is completely unsettling, right? And I have twins, and I know exactly what parents mean when they say they don't want to get their kids vaccinated. But the reality is that is not what is leading to a child's autism. That is actually maybe even protecting them. There have been studies that have looked at the COVID vaccine and whether or not the COVID vaccine causes autism, and it doesn't, but it will prevent you getting COVID. So I think that for whatever reason, because you're taking your child to the pediatrician and you're injecting something in them, it's very scary for parents to think that this potential compound could be triggering something. But the science is very, very clear about this. There's more science around studying vaccines and autism than there is under any other environmental factor. And that's a real shame, because there are a number of different potential hypotheses that have been ignored in autism because we have been so focused on addressing whether or not vaccines cause autism.
This has been a really fascinating conversation. I appreciate it. As we are nearing the end of today's webinar with our last set of questions, let's first talk about early support. So, Dr. DiCicco-Bloom, does brain development research support the idea of critical windows for intervention?
So the answer, of course, is yes and no. There are beautiful studies, particularly in animal models, where we know that different parts of the brain develop at different times. And associated with that, using simple things like a mouse or a rat, we can have behaviors that we can assign to different regions. So let me take two obvious ones. For a mouse, being social is going over to another mouse and smelling them everywhere, face, backside, and interacting with them. Another behavior a mouse can do is it can groom itself, clean its fur, or move up and down. And so what we just talked about is a mouse version of a social interaction or a repetitive behavior. And so, for example, in a particular genetic form of autism called tuberous sclerosis syndrome, you can see abnormalities in these behaviors. And what you can find is that when you have that mouse, you can block the abnormality with a drug. And in that case, if you give the drug early, you can reverse the social and repetitive motor behaviors. If you give it closer to adolescence, you can only reverse the social, but not the repetitive behaviors. And so those are examples in the very convenient animal model universe that allow us to think of it as a window when something forms and when it might be amenable to interaction. But I think another way to think about this is we talked about the brain being plastic. That is, it can adapt, it can change. And one of the areas that research is very encouraging, as Dr. Manzini spoke about, is understanding which genes may have effects on certain regions. And by manipulating the gene or manipulating a cellular signal that's regulated by that gene, we can address some of those. And in these single gene causes like tuberous sclerosis or Fragile X syndrome, there have been very useful clinical studies examining what medications could alter either the gene itself or downstream cell signaling and lead to improvement in some of those individuals. So I think there are critical windows, but I will tell you as a clinician, when I learned about the eyeball and from ophthalmology studies, it was said, based on a lot of significant animal models at every level, that the eyes make connections from the retina to the thalamus to the cortex. And once you've made your visual cortex, it can't be helped. So once it's messed up, can't intervene. And then about 10 years ago at the Society for Neuroscience, there were major reports in clinical studies that indicated we could open up this critical window, this restriction, because basic research found that some of this restriction was due to cells and the proteins they make. And by using medications, they could interfere with those proteins which closed that window and opened it up. This is nothing you could do in a person. This was something that was quite a surprise within the field. We're talking about decades of research, talking about rigid critical windows. I think we need to be a little bit more optimistic and consider trying to understand how to open those up for therapeutic intervention.
Thank you so much. Dr. Manzini, for families seeking clarity through testing, if a genetic difference is identified, why doesn't that usually lead to a specific medical treatment?
So in this case, there has been a lot of research to try and develop therapeutics for specific genetic causes, right? The Fragile X syndrome gene was identified in 1991, and there have been multiple clinical trials trying to figure out if particular drugs could be helpful for this condition. There is one that's happening right now. And the initial ones failed for a variety of reasons, but they also really taught us how to run clinical trials for autism and intellectual disability and neurodevelopmental conditions. You know, I also work on muscle disease. That have been a field where there's been a lot of success with gene therapy, but in a way, the readout is easier. Do the kids walk further than they walked before? You get a number. With autism has been more complicated. And unfortunately, also because there are hundreds of genes, you will have some genes where you have hundreds of labs working on, and you will have genes where you have 5 labs around the world. What I always like to tell parents, like even if you have an obscure genetic mutation, there is likely somebody around the world that is working on that gene. So if you want, reach out to them to try and figure out what is going on. But I was making the example of muscular dystrophy. Differently from the muscle, the brain is very difficult to access. And also it functions at this very specific set point. So even doing gene therapy, where you would restore that one specific gene, you cannot put too much, you cannot put too little, because you risk not solving the problem or causing additional problems. And there are a lot of labs right now, a lot of scientists that are trying to figure that out. The brain is very important. It's difficult to access. It needs to be protected at all costs, because if it doesn't work, then nothing works. And so we have to be very careful about going into and manipulating it.
Okay, thank you. And for our final question of the day, Dr. Halladay, what has environmental research helped us understand about prevention versus prediction?
Yeah, so that's a really good question. So I think that it depends on the environmental factor. So far, we have not been able to change the prevalence of autism like we did, like the example that Dr. DiCicco-Bloom mentioned, lead. We were able to take lead out of gasoline, take lead out of paint, and saw an increase in cognitive abilities and a decrease in lead poisoning. That's a major public health success story. We have not been able to do that with autism yet. We have tried to eliminate different exposures in the environment. For example, there were certain concerns about different components of vaccines that were taken out. That hasn't shown to have an effect at all. So far, we have not been able to change the prevalence of autism through any sort of modification of the environment, which shows you how complex autism is and how driven it is by genetics. So I think that environmental factors, we definitely can hope to change the prevalence of autism. There are things that pregnant women should be doing anyway. You should be washing your hands. You should be staying away from sick people. You should be getting vaccinated so that you don't get the flu during pregnancy. You should avoid air pollution. You should try not to use toxic pesticides. You should be able to live in an environment that you have access to great services and you can get services for your child with autism easily. We hope these for all people, but they're not something that people obviously always have control over. So I think that we're moving towards prevention. We haven't gotten there yet, but we definitely have reached prediction. We can definitely say that certain environmental factors lead to a higher probability of having an autism diagnosis. And what that definitely means is if you are exposed to these things, if this maternal health condition is with you, if you've experienced these things, that you should continue to get closely monitored by your pediatrician or even get a developmental pediatrician who will look for things like autism and ADHD or other developmental concerns. So we haven't really gotten to prevention yet, but that's definitely an area that we kind of want to think about, at least preventing the more severe or detrimental parts of autism.
Thank you. I want to sincerely thank Drs. DiCicco-Bloom, Manzini, and Halladay for returning to this conversation and for engaging so thoughtfully with the questions that came from this community. I also want to thank the NJACE for being co-sponsor for this event. And lastly, I want to thank all of you, not just for attending today, but for continuing to ask careful, informed questions. We have made note of all the questions that you have asked, and we will add them to our ongoing database of causes-related questions for future sessions. Also, please know that this webinar has been recorded and will be made available for all to watch. This webinar is part of an ongoing dialogue, and we're grateful to be part of it with you. Thank you for joining us.